The study of the genetics and archaeogenetics of the ethnic groups of South Asia aims at uncovering these groups' genetic history. The geographic position of India makes Indian populations important for the study of the early dispersal of all human populations on the Eurasian continent. Our studies based on mtDNA variation have reported genetic unity across various Indian subpopulations. Conclusions of studies based on Y-Chromosome Variation and Autosomal DNA Variation have been varied. Although many researchers argue that most of the ancestral nodes of the phylogenetic tree of all the mtDNA types originated in the subcontinent, recent genome studies appear to show that most South Asians are descendants of two major ancestral components, one restricted to South Asia and the other component shared with Central Asia, West Asia and Europe. It has been found that the ancestral node of the phylogenetic tree of all the mtDNA types typically found in Central Asia, West Asia and Europe are also to be found in South Asia at relatively high frequencies. The inferred divergence of this common ancestral node is estimated to have occurred slightly less than 50,000 years ago. In India, the major maternal lineages, or mitochondrial DNA haplogroups, are M, R and U, whose coalescence times have been approximated to 50,000 BP. The major paternal lineages represented by Y chromosomes are haplogroups R1A1, R2H, L and J2. Many researchers have argued that Y DNA haplogroup R1A1 is of autochthonous Indian origin. However, proposals for a Central Asian origin for R1A1 are also quite common. Overview. All the mtDNA and Y chromosome lineages outside Africa descend from three founder lineages, M, N and R haplogroups for mtDNA and C, D and F haplogroups for the Y chromosome. All these six founder haplogroups can be found in the present-day populations of South Asia. Moreover, the mtDNA haplogroup M and the Y chromosome haplogroups C and D are restricted to the area east of South Asia. All the West Eurasian populations derive from the N and R haplogroups of mtDNA and the F haplogroup of the Y chromosome. Endicott et al. State that these facts are consistent with the hypothesis of a single exodus from East Africa 65,000 years ago via a southern coastal route, with the West Eurasian lineages separating from the South Asian lineages somewhere between Northeast Africa and South Asia. mtDNA. The most frequent mtDNA haplogroups in the Indian subcontinent are M, R and U, arguing for the longer-term rival Y chromosome model. Stephen Oppenheimer believes that it is highly suggestive that India is the origin of the Eurasian mtDNA haplogroups, which he calls the Eurasian Eves. According to Oppenheimer, it is highly probable that nearly all human maternal lineages in Central Asia, the Middle East and Europe descended from only four mtDNA lines that originated in South Asia 50,000–100,000 years ago. Macrohaplogroup M. The macrohaplogroup M, which is considered as a cluster of the Proto-Asian maternal lineages, represents more than 60% of Indian mtDNA. The M macrohaplotype in India includes many subgroups that differ profoundly from other sublineages in East Asia, especially Mongoloid populations. The deep roots of M phylogeny clearly ascertain the relic of Indian lineages as compared to other M sublineages, suggesting in situ origin of these subhaplogroups in South Asia, most likely in India. These deep-rooting lineages are not language-specific and spread over all the language groups in India. Virtually all modern Central Asian mtDNA lineages seem to belong to the Eastern Eurasian rather than the Indian subtypes of Haplogroup M, which indicates that no large-scale migration from the present Turkish-speaking populations of Central Asia occurred to India. The absence of Haplogroup M in Europeans compared to its equally high frequency among Indians, Eastern Asians and in some Central Asian populations contrasts with the Western Eurasian leanings of South Asian paternal lineages. Most of the extant mtDNA boundaries in South and Southwest Asia were likely shaped during the initial settlement of Eurasia by anatomically modern humans. Macrohaplogroup R. The macrohaplogroup R is also widely represented and accounts for the other 40% of Indian mtDNA. A very old and most important subdivision of it is haplogroup U that, while also present in West Eurasia, has several subclades specific to South Asia. Most important South Asian haplogroups within are haplogroup U. Haplogroup U is a sub-haplogroup of macrohaplogroup R. The distribution of haplogroup U is a mirror image of that for haplogroup M. The former has not been described so far among Eastern Asians but is frequent in European populations as well as among Indians. Indian U lineages differ substantially from those in Europe, and their coalescence to a common ancestor also dates back to about 50,000 years. Y-Chromosome. The major Y-chromosome DNA haplogroups in the subcontinent are haplogroup F's descendant haplogroups are L, H and J. The South Asian Y-chromosomal gene pool is characterized by 5 major lineages, R1A, R2H, L and J2. The geographical origins are listed as follows, according to the latest scholarship. Haplogroup L India. Haplogroup L shows time of Neolithic expansion. The clade is present in the Indian population at an overall frequency of California, 7 to 15%. There are 3 sub-branches of haplogroup L and all 3 are found mostly in South Asia. Haplogroup L has higher frequency among South Indian castes and reaches up to 68% in some castes in Karnataka but is somewhat rarer in North Indian castes and reaches up to 38% in some castes in Gujarat. They make a case for an indigenous origin of LM76 in South Asia as the spatial distributions of both LM76Hg frequency and associated microsatellite variants show a pattern of spread emanating from southern India. The presence of haplogroup L is quite rare among tribal groups. Pakistan. Haplogroup L3 is found frequently among Burusha and Pashtuns. Its highest frequency can be found in southwestern Balochistan province along the Makran coast to Indus River Delta. L3A is found in approximately 23% of Nuristani in northwest Pakistan. The clade is present in moderate distribution among the general Pakistani population. Haplogroup H. Haplogroup H is found at a high frequency in South Asia. H is rarely found outside of the South Asia but is common among the Romanis, particularly the HM82 subgroup. Haplogroup H is frequently found among populations of India, Sri Lanka, Nepal, Pakistan and Maldives. All 3 branches of haplogroup H are found in Indian subcontinent. It is a branch of haplogroup F and descends from GHIJK family. Haplogroup H is believed to have arisen in South Asia between 30,000 and 40,000 years ago. Its probable site of introduction is South Asia, since it is concentrated there. It seems to represent the main Y-chromosome haplogroup of the Paleolithic inhabitants of Indian subcontinent. Some individuals in South Asia have also been shown to belong to the much rarer subclade H3. Haplogroup H is by no means restricted to specific populations. For example, H is possessed by about 28.8% of Indo-Aryan castes, and in tribals about 25 to 35%. Haplogroup R2 in South Asia, the frequency of R2 and R2A lineage is around 10 to 15% in India and Sri Lanka and 7 to 8% in Pakistan. At least 90% of RM124 individuals are located in the Indian subcontinent. It is also reported in Caucasus and Central Asia at lower frequency. India, it is found among people of West Bengal at 23%, Hindus from New Delhi at 20% and Bania from Bihar at 36%. Among tribal groups, Lodhas of West Bengal show it at 43%, while Bhil of Gujarat at 18%. Chenchu and Palan of South India at 20% and 14% respectively. Tharo of North India shows it at 17%. It is also significantly high in many Brahmin groups, including Punjabi Brahmins, Bengali Brahmins, Bengali Kasthas, Kinganista Brahmins, Chaturvedas Bhargavas, Kashmiri Pandits and Lingayut Brahmins. North Indian Muslims have a frequency of 11% and 9%, while Dawoodi Bohra Muslim in the western state of Gujarat have a frequency of 16% and Mapila Muslims of South India have a frequency of 5%. This lineage also forms 5% of Punjabi males. Pakistan, the R2 haplogroup is found in 14% of the Burusho people. Among the Hunza, it is found at 18%, while the Parsis show it at 20%. It is also found in the northeastern part of Afghanistan. Sri Lanka, 24.1% of the Sinhalese of Sri Lanka are found to have R2 as subclades of haplogroup P according to the research of Dr. Tumas Kivizald published in The Genetics of Language and Farming Spread in India. Sample size used was 87 subjects. Maldives, 13% of the Maldivian people of Maldives are found to have R2. Nepal, in Nepal, R2 percentages range from 2% to 26% within different groups under various studies. Newas show a significantly high frequency of 26%, while people of Kathmandu show it at 10%. Haplogroup R1A1 in South Asia. R1A1 has been observed often with high frequency in a number of demographic groups, which leads some to see it as the locus of origin. According to Pamjav et al., Inner and Central Asia is an overlap zone for the R1A1Z280 and R1A1Z93 lineages, implies that an early differentiation zone of R1A1M198 conceivably occurred somewhere within the Eurasian steppes of the Middle East and Caucasus region as they lie between South Asia and Eastern Europe. Underhill et al. Concluded that R1A1A1, the most frequent subclade of R1A, split into R1AZ282 and R1AZ93 at circa 5800 before present in the vicinity of Iran and eastern Turkey. According to Underhill et al., his suggests the possibility that R1A lineages accompanied demic expansions initiated during the Copper, Bronze, and Iron Ages. Quote, they further note that the diversification of Z93 and the early urbanization within the Indus Valley also occurred at this time, and the geographic distribution of R1AM780 may reflect this. Posnick et al. Note that striking expansions occurred within R1AZ93 at 4500–4000 years ago, which predates by a few centuries the collapse of the Indus Valley civilization. Mascarenas et al. Note that the expansion of Z93 from Transcaucasia into South Asia is compatible with the archaeological records of eastward expansion of West Asian populations in the 4th millennium BCE, culminating in the so-called Kura-Araxes migrations in the post-Auric IV period. India. In India, high percentage of this haplogroup is observed in West Bengal Brahmins to the east, Kinganista Brahmins to the west, Khatris in north and Iyengar Brahmins of south. It has also been found in several South Indian Dravidian-speaking tribals including the Chanchu and Valmikis of Andhra Pradesh as well as the Yadav and Kala of Tamil Nadu, suggesting that M17 is widespread in these southern Indians tribes, and 47% in Punjab. Pakistan. In Pakistan it is found at 71% among the Mohana of Sindh province to the south and 46% among the Baltis of Gilgit-Baltistan to the north. Sri Lanka. 23% of the Sinhalese people were found to be R1A1A positive according to the research of Dr. Tumas Kivizald published in The Genetics of Language and Farming Spread in India. Sample size used was 87 subjects. Maldives. In Maldives, 24% of the Maldivian people were found to be R1A1A positive. Nepal. People in Terai region, Nepal show R1A1A at 69%. Haplogroup J2. Haplogroup J2 reflects presence from Neolithic period in the subcontinent. The frequency of J2 is higher in South Indian castes than in North Indian castes or Pakistan. Haplogroup J2 frequency is higher among South Indian middle castes at 21%, followed by upper castes at 18.6% and lower castes 14%. J2 is present in tribals too and has a frequency of 11% in Austroasiatic tribals. Among the Austroasiatic tribals, the predominant J2 occurs in the Loda. J2 is also present in the South Indian hill tribe Toda at a frequency of 38.46% and in the Khel tribe of Uttar Pradesh at a frequency of 33.34%. It is also found at 3% among the Sadis. In Maldives, 22% of Maldivian population were found to be haplogroup J2 positive. Reconstructing Indian population history. The Indian Genome Variation Consortium divides the population of the subcontinent into 4 linguistic groups: Indo-European, Dravidian, Tibeto-Burman and Austroasiatic. The molecular anthropology studies use 3 different types of markers: mitochondrial DNA variation, which is maternally inherited and highly polymorphic; Y-chromosome variation, which involves uniparental transmission along the male lines; and autosomal DNA variation, mtDNA variation. Most of the studies based on mtDNA variation have reported genetic unity of Indian populations across language, caste and tribal groups. It is likely that haplogroup M was brought to Asia from East Africa along the southern route by earliest migration wave 60,000 years ago. According to Kivizald et al., minor overlaps with lineages described in other Eurasian populations clearly demonstrate that recent immigrations have had very little impact on the innate structure of the maternal gene pool of Indians. Despite the variations found within India, these populations stem from a limited number of founder lineages. These lineages were most likely introduced to the Indian subcontinent during the Middle Paleolithic, before the peopling of Europe and perhaps the Old World in general. Basu et al. Also emphasizes underlying unity of female lineages in India. Y-chromosome variation. Conclusions based on Y-chromosome variation have been more varied than those based on mtDNA variation. While Kivizald et al. Proposes an ancient and shared genetic heritage of male lineages in India, Bamshad et al. suggests an affinity between Indian male lineages and West Eurasians proportionate to caste rank and places caste populations of southern Indian states closer to East Europeans. Basu et al. Concludes that Austroasiatic tribal populations entered India first from the northwest corridor and much later some of them through northeastern corridor, whereas Kumar et al. Analyzed 25 Indian Austroasiatic tribes and found strong paternal genetic link among the sublinguistic groups of the Indian Austroasiatic populations. Mukherjee et al. Places North Indians between West Asian and Central Asian populations, whereas Cordo et al. Argues that the Indian caste populations are closer to Central Asian populations. Sahu et al. And Sengupta et al. Suggest that Indian caste populations have not been subject to any recent admixtures. Sanghamitra Sahu concludes his study with, "It is not necessary, based on the current evidence, to look beyond South Asia for the origins of the paternal heritage of the majority of Indians at the time of the onset of settled agriculture. The perennial concept of people, language, and agriculture arriving to India together through the Northwest Corridor does not hold up to close scrutiny. Recent claims for a linkage of haplogroups J2L, R1a, and R2 with a contemporaneous origin for the majority of the Indian caste as paternal lineages from outside the subcontinent are rejected, although our findings do support a local origin of haplogroups F** and H. Of the others, only J2 indicates an unambiguous recent external contribution from West Asia rather than Central Asia. The current distributions of haplogroup frequencies are, with the exception of the lineages, predominantly driven by geographical, rather than cultural determinants. Ironically, it is in the northeast of India, among the TB groups, that there is clear-cut evidence for large-scale demic diffusion traceable by genes, culture, and language, but apparently not by agriculture. Autosomal DNA variation. Results of studies based upon autosomal DNA variation have also been varied. In a major study using over 500,000 biallelic autosomal markers, Reich hypothesized that the modern Indian population was the result of admixture between two genetically divergent ancestral populations dating for the post-Holocene era. These two reconstructed ancient populations he termed ancestral South Indians and ancestral North Indians. According to Reich, our ancestry is significantly higher in Indo-European than Dravidian speakers, suggesting that the ancestral ASI may have spoken a Dravidian language before mixing with the Ani. Further building on Reich et al.'s characterization of the South Asian population as historically based on admixture of Ani and ASI populations, a 2011 session paper by Maulucioni et al. States that a major Ani-ASI mixture occurred in the ancestors of both northern and southern Indians 1,200–3,500 years ago, overlapping the time when Indo-European languages first began to be spoken in the subcontinent. Basu et al. Concludes that Dravidian tribals were possibly widespread throughout India before the arrival of the Indo-European-speaking nomads, and that formation of populations by fission that resulted in founder and drift effects have left their imprints on the genetic structures of contemporary populations. The geneticist P. P. Majumdar has recently argued that the findings of Reich et al. Are in remarkable concordance with previous research using mtDNA and Y-DNA. Central Asian populations are supposed to have been major contributors to the Indian gene pool, particularly to the northern Indian gene pool, and the migrants had supposedly moved into India through what is now Afghanistan and Pakistan, using mitochondrial DNA variation data collated from various studies. We have shown shown that populations of Central Asia and Pakistan show the lowest coefficient of genetic differentiation, with the North Indian populations, a higher differentiation with the South Indian populations, and the highest with the Northeast Indian populations. Northern Indian populations are genetically closer to Central Asians than populations of other geographical regions of India. Consistent with the above findings, a recent study using over 500,000 biallelic autosomal markers has found a north-to-south gradient of genetic proximity of Indian populations to Western Eurasians. This feature is likely related to the proportions of ancestry derived from the Western Eurasian gene pool, which, as this study has shown, is greater in populations inhabiting northern India than those inhabiting southern India. Genetic distance between caste groups and tribes. Studies by Watkins et al. And Kivizald et al., based on autosomal markers, conclude that Indian caste and tribal populations have a common ancestry. Reddy et al. Found fairly uniform allele frequency distributions across caste groups of southern Andhra Pradesh, but significantly larger genetic distance between caste groups and tribes, indicating genetic isolation of the tribes and castes. Viswanathan et al., in a study on genetic structure and affinities among tribal populations of southern India, concludes genetic differentiation was high and genetic distances were not significantly correlated with geographic distances. Genetic drift therefore probably played a significant role in shaping the patterns of genetic variation observed in southern Indian tribal populations. Otherwise, analyses of population relationships showed that all Indian and South Asian populations are still similar to one another, regardless of phenotypic characteristics, and do not show any particular affinities to Africans. We conclude that the phenotypic similarities of some Indian groups to Africans do not reflect a close relationship between these groups, but better explained by convergence. A 2011 study published in the American Journal of Human Genetics indicates that Indian ancestral components are the result of a more complex demographic history than was previously thought. According to the researchers, South Asia harbors two major ancestral components, one of which is spread at comparable frequency and genetic diversity in populations of Central Asia, West Asia and Europe. The other component is more restricted to South Asia. However, if one were to rule out the possibility of a large-scale Indo-Aryan migration, these findings suggest that the genetic affinities of both Indian ancestral components are the result of multiple gene flows over the course of thousands of years. Bibliography.